our channels. So today we are discussing about instrumental in microbiological lab. Instruments in microbiology lab. Today we will discussing about it. In this video, spokes on the different instruments they can they can features in different instruments, especially in different environmental also used. Most critical instruments in a microbiology laboratory are the microscope for visualizations, the autoclave for sterilizations, and the incubator for growing microbial cultures. Because microbiology it requires a sterile environment to isolate and analyze microscopic life. This is the observations and analysis of microscopic techniques. So the essential microbiology laboratory instrument has so many categories are given below. So incubators also. Incubators so providers and insulated temperatures controlled environment, typically 37 degrees centigrade also. Okay, next here autoclave. Autoclave it generates high pressure steam at 121 degrees centigrade to sterilize growth media and glassware and hazardous bio waste by degenerating microbial proteins also. So autoclave, this is also the sterilizations and biosafety instruments also. So autoclave. And otherwise autoclave normally 121 degrees C and 15 minutes also. And next one Bunsen burner. So this is the Bunsen burner. This is the it will be connected to a gas supply to produce an open flame. It provides us direct sterilizations for inoculating tools and maintenance in a small sterile upgrade zone on the open bench. So next here, the anaerobic jar also. It is a shielded container also, and that chemically removes oxygens, it allowing the selective cultivation of anaerobic bacteria. So what are that anaerobic bacteria already anaerobic bacteria? So what are that? So anaerobic means for anaerobic applicate anaerobes to kill the strictly inhibited by oxygens. So low oxygen environments to survive the anaerobic microorganisms that survive the grow and replicate without oxygens for anaerobic bacteria without oxygen. It will be exposed to molecular oxygen. It can be toxic or lethal. Too many of these species are required. Okay, so different type of the anaerobes, applicate anaerobes and facultative anaerobes and aero tolerant anaerobes also. And next slides are pure culture techniques. So pure culture techniques is a single culture colonies also isolated. So microbiologically cultured media for pure culture techniques is a very important. So it will be the survival and growth of microorganism depend on available nutrients and favorable growth environments also there. So the nutrient preparations that are used for culturing microorganisms are called media. So media for different cultural media also. So the pure cultural media different types. So what are the types? Solid media and liquid media and semi-solid media. The pure culture media is a specialized nutrient solutions used in microbiology to isolate, grow and maintain a single species of microorganisms from a mixed populations also. So the media it will be used for pure culture techniques is very essentially. Okay. So solid media to prepare adding an agent like agar. So and liquid media is a broth also. Liquid media is a broth. And semi-solid media, it contains the minimal amount of agar. So enrichment media it is supplemented with highly nutritious substances like blood serum or egg yolk, isolating the organisms. For blood agar is a common. And next slide for physical forms. 3 physical forms also. What are that? So there are the temperature, pH, and so physical form maintaining to the semi-solid medium. So semi-solid medium, it contains the minimal amount of agar, approximately 0.2% to 0.5%. This is the semi-solid media. It is primarily used to test cell motilities, and it can be used fermentations studies. So semi-solid media is a fermentation studies. What can you use for semi-solid media? First of all, agar plates and agar deep tube cultures, and third one, this is the broth cultures and agar slants. This is the semi-solid agar plates and tubes, test tubes also. So what are the functions and used for and purpose of media? So general purpose media, it contains the nutrients. What are the nutrients are present in peptone and beef extract that support non-fastidious bacteria. For example, nutrient agar. Okay. These slides, next slide, the shape of bacteria and margins. What are that margins and size? So many size. So included the so what are the types and shapes, margins and evaluations also identified. So next slide, microscope. So microscope are mainly important for the microbiologically very important. So previously I told you, so what are the microscope? What is it microscope? Also essentially important. I told you. And types of microscopy I told you. So compound light microscope, it is used a multi-lens systems and visible light to magnify bacteria, fungi and protozoa also. It is vital for observing cell morphology. It will be arrangement and staining characteristics. So microscopic terms. So small naked eye to visualizing the bacteria also. So what are the microscope and what microscopic terms? So magnifications. Magnification is essentially very important. Microscopic terms. So magnifications. What is it magnifications? The scale of enlargement. That's all also. The scale of enlargement. Magnification is the scale of enlargement. And second terms, resolutions. Resolutions. Already told you. And the next slides. So the types of microscopy clearly explaining about what are the terms involved in microscopic detailed explanations I give you there. Please one by one view that each one slides. Okay. So resolutions, what is it? The clarity and details. And next terms, this is also the light microscopic. View the pictures also light microscopic. So you see, very important for the various component that direct light to your eyes of a digital sensor. These also the so microscopically, what are the compound microscope? Very essentially you could be identified. So first of all, the terms resolutions, numerical aperture, refractive index, working distance, field of view, depth of field. These also the microscopic terms. This is the another one, optical properties. So numerical aperture, the dimensionless number that measures a lens ability. Okay, that measures a lens abilities also. And next one, microscopic parts. What are that? Ocular lenses. So ocular lenses, the very important microscopically structure and mechanical components. Ocular lens, eye piece. Ocular lens, what is it? Eyepiece. That's all. The lens at the top of the microscope. The lens at the top of the microscope that you look through typically provides an independent 10x magnifications. Okay. Next, objective lens. Objective lens. This is the primary lenses. Objective lenses, primary lenses closest to the specimen, mounted on a rotating nose piece. Mounted on a rotating nose piece. Standard options include scanning 4x. 4x means low power, 10x high try, and 40x, and oil immersion 100x. Okay. And next slide also. So slide is a piece of glass upon which the specimen is placed for viewing. So specimens viewing. This is the very important for slides. So adjustment to the nap. So this is the very important for slides. And next is a cover slip. So cover slip for thin small piece of glass or plastic used to hold the specimens. Cover slip for mainly very important and thin slides, thin or flattened structure and transparent piece of material, and typically high quality borosilicate glass or plastic placed over a specimen on a microscopic slides. And next, diaphragm lever. Diaphragm lever, this is the disc device. So disc device also. So it located beneath the stage. It controls the amount of light passing through. This is a small adjustable amp. So diaphragm lever it is a small adjustable amp. Okay, next, substage light. It's source of light which passes through specimens.
substage light passes through specimens. And next, so substage light also. And next, base. So bases, bottom stand also, which houses the light. So base is a structural stability also. It is heavily weighted for it to prevent the microscopic for vibrating and shaking and focusing at high. The base also. So the arm basic frame. Arm is a basic frame. It used to carry microscope. It's a very important for arm is a structural supported, and it hold the nose piece and objective lenses and eyepiece assemblies is very important also. And next, thumb clip. What is a thumb clip? So thumb clip is a hold slide on stage to prevent it from slipping around. So more commonly known as the stage clip. Another one named slide holder clump. Slide holder clump, CLAMP, is the spring loaded arm. It located the top of your microscopes. Okay. And next, horse adjustment nap. Horse adjustment nap. So it roughly focuses images. Okay. Focuses images. And next slides, fine adjustment nap. So fine adjustment nap. So it is a sharpness images of the specimens. Sharpness adjustment of the specimens also. So it moving the specimens and objective lenses very small distances. So smaller distances it located and directly inside or on top of the larger adjustment nap of the microscope arm. And this the high power necessities. High power necessity, what is it? 40x high power and 100x oil immersion objective lenses are used. Fine adjustment nap also. And next, mechanical stage control. Mechanical stage control. So what are that? We view the slides. Mechanical source control nap or a pair of coaxial tiles. It exists in the x-axis, y-axis. So what is it? X-axis is a left and right also. Okay, x-axis left and right. You see the pictures. Okay, and next y-axis is a forward and backward with high precisions. So this is the top knob and bottom knob. Top knob otherwise y-axis and bottom knob always x-axis also. This is the, you see the pictures, and y-axis is the forward and backward, x-axis left and right. This is the knob works also. So you see the pictures. How do you visualize the microscopically pictures also? So manually push the slide with your fingers, which is too erratic under the high magnifications. So the controls are